Hello students. So today we are going to discuss with few important questions of the chapter. This chapter is about to be end. We have discussed about different kinds of cell organelles in plants, animals, transpiration, how it happens, how respiration happens, excretory organs, and what are the internal cell organs, functions of internal cell organs, plants and animals. So now we are going to discuss few important questions. Can you name two cell organelles we have studied that contain their own genetic material? Mitochondria and plastids are the two cell organelles that contain their own genetic material. Both these organelles have their own DNA and ribosomes. We have discussed in few previous videos. Make a comparison, write down ways in which plant cells are different from animal cells. Animal cells are generally small in size. In animal cells, cell wall is absent. In animal cells, except the protozoan Euglena, no animal cell possesses plastids. And vacuoles also smaller in size in animal cells. So in animal cells what happens is basically they are smaller in size, cell wall is absent, and no animal cell possesses plastids except there is an exception that protozoan Euglena, it has plastids. Rest they don't have plastids, and vacuoles are smaller in size in animal cells. In plant cells it is very usually larger in size. In this, cell wall is present. In plastids, that is chromoplast and leucoplast, which helps in the pigment formation, are also present, and vacuoles are larger in size. What would happen to the life of a cell if there is no Golgi apparatus? Membranes of Golgi apparatus are often connected to ER membranes, that is endoplasmic reticulum membranes. It collects simpler molecules and combines them to form more complex molecules. So what Golgi apparatus does is, Golgi apparatus is connected to endoplasmic reticulum membranes, and what it does it collects the simpler molecules and converts it into complex molecules, and these are often then packaged in small vesicles and are either stored in the cell or sent out as per the requirement. Thus, if the Golgi apparatus is absent in the cell, then the ever process of storage, modification, packaging of products will not be possible. So what happens is, it first is converts the simpler, it absorbs the collects the simpler molecules and converts it into complex molecules, and then it also convert them into small packets of vehicles, and they are either stored in the cell or are sent out as per the requirement. So if Golgi apparatus are not present, but it will first, what will happen that those materials will be not act as it. There will be no storage or modification or packaging of this nutrients products. The formation of complex sugars from the simple sugars will not be possible, as it takes place with the help of the enzyme present in Golgi bodies. So we have discussed in a few classes before that complex sugars are form simple sugars with the help of the enzymes present in Golgi bodies. The Golgi apparatus is involved in the formation of lysosomes. Thus, if the Golgi body is absent in the cell, the synthesis of lysosomes will not be possible in the cell. So the Golgi apparatus is involved in the formation of lysosomes. So if the Golgi apparatus is absent, synthesis of lysosomes will not happen. Which organelle is known as a part of the cell? Why? Mitochondria. We have discussed this earlier. Mitochondria is known as a part of the cell. Mitochondria creates energy for the cell, and this process of creating energy for the cell is known as cellular respiration. Most chemical reactions involved in cellular respirations occur in the mitochondria. So it basically is known as a part of the cell. It creates energy for the cell, and the process of creating energy for the cell is known as cellular respiration. Most chemical reactions involved in cellular respiration occur in mitochondria. The energy required for various chemical activities needed for life is released by mitochondria in the form of adenosine triphosphate. Where do lipids and proteins constituting the cell membrane get synthesized? Lipids and proteins constituting the cell membrane are synthesized in ER, that is endoplasmic reticulum. We have discussed about smooth endoplasmic reticulum and of endoplasmic reticulum. How does an amoeba obtain its food? We have discussed about forming a layer across the particle, that is the process known as the endocytosis, with its membrane, which is flexible membrane. The flexibility of the cell membrane enables the cell to engulf the solid particles of the food and other materials from its external environment. So what does amoeba does is, amoeba creates or covers the particles around itself, around its body, and that process is known as endocytosis. What is osmosis? Osmosis is the process of movement of molecules from a region of higher concentration to a region of lower concentration through a selectively permeable membrane. So this was all about the session of cell. Thank you.